Hello, good morning, and welcome to all of you. This is Rajesh Kumar Singhal, and I am here to present a new lecture on the physiology of flooring part 2. So, friend, this is very important topic for all the competitive exams like CSIR, ICR, and other life sciences exams. And in the first part of this lecture, like physiology of flooring part first, we have discussed a lot of points regarding the physiology of flooring, such as we have discussed the introduction of floor, then we have discussed the structure of floors, and in this we have discussed about the carpal, sepal, petal, stamen, also megagametophyte, microgametophytes. We have discussed about the details in the structure of flower. Then we have discussed about the life cycle of angiosperm, that how the double fertilization take place and how the formation of endosperms take place. Then we have discussed about the evolution of angiosperm, that how the angiosperms are evolved from the primary gymnosperm or primary basal angiosperms. Then we have discussed about the monocarpic and polycarpic habits of plant, that how the monocarpic plants convert their whole stem into the reproductive stem. Then we have discussed about the phase changes, that how the changes take place from juvenile to vegetative, vegetative to vegetative, reproductive, vegetative to vegetative, reproductive. Then we have discussed several factors which affect the phase changes like plant age. Then we have discussed about the some genes, principal genes, which are regulates the flowering process. And at the end of the lecture, we have discussed about the ABC model, that how the different genes regulate the formation of sepal, petal, carpel, and stamen. So in the last lecture, we have discussed some genes which are involved in the flowering process, such as agamus, Apatella 1 and 2, CCA circadian clock associate 1, CO constant, ELF 3 and 4, early flowering 3 and 4, then FCA, FLC, flowering locus C, flowering locus T, gibberellic acid 1, lumini dependence, leafy, talk 1, that is timing of cap 1, vernalization 1, 2, 3. So from this slide, the main conclusion is that there are number of genes which are involved during the flowering process. And other conclusion is that there are number of pathway, like there is one circadian clock pathway, then second one is the long day pathway, then there will be a autonomous pathway, then there will be a gibberellic acid pathway, then there will be a vernalization pathway. So friends, in the flowering process, many of these pathways are regulated. Means these processes are integrated and regulated the flowering process. Also from these slides, we can say there are number of genes which act as a flowering repressor, and some genes act as a floral promoter. So in this lecture, we will discuss about these pathways, details in about these pathways. So in this lecture, we will discuss about some flowering old theories, then what is modern theory, then we will discuss in detail about circadian rhythms, then we will discuss about the phototropism, photoperiodism and thermoperiodism, then vernalization, then autonomous pathway, then we will discuss about GA pathway, then how the environment regulate the flowering process, then regulatory gene in flowering pathway and their functions, Then we will also discuss about the signaling pathway associated during the flowering process. We will also discuss the crosstalk of different pathway during the flowering process. So these are the let's start our lecture with the flowering theory. So there are number of theory which are proposed for the flowering process. So first theory is the CN relationship theory. This is carbon nitrogen relationship theory. This theory is given by the Kells 1913, and according to this theory, the high CN ratio promote the flowering. So what this theory says? This theory says that high ratio of carbon to nitrogen promote the flowering. Then there will be a second theory, that is trace element nutrient theory. So this theory proposed by the Hillman in the lemma. So what this theory say? According to this theory, the copper and iron, which are trace elements, promote the flowering in the lemma plant. Then third theory is the water stress theory. So what this theory says? This theory says that zero induction. What is the zero induction? Means drought or water stress induce the flowering in the chenopodium. Chenopodium or Chicorium. So in this, the water stress promote the flowering. And the next theory is the Florism theory. So what this theory says? According to this theory, there will be a hypo, there will be a hypothetical molecules hormone which triggers the flowering, and this is known as florigen. Name is given by the scientist Mel Chela Khan in 1937, and these hormones are produced in the leaves and acts in the shoot apical meristem of growing buds and tips. And this florism is identical to the flowering locus T. Also the this florism is regulated by the anti-florism. So what is this anti-florism? So these are also hormones which counteract the function of florism, and the example is the TFL1 in Arabidopsis, that is also known as terminal terminal flower 1. And in tomato, it's SP1, that is self pruning 1. So this is the your florism theory, which is given by the stack 37. Then the fifth theory is the flower inhibition theory, proposed in the 1950 by the scientist Lona. So according to this theory, the unfavorable conditions such as drought, heat, cold inhibit the flowering process. And the next theory is the plant age theory. We also discussed in the last lecture that plant have a phase, means they have definite phase like vegetative, reproductive. So they have reproductive phase. In this they form the competent and determined buds. So according to plant age, there will be a formation of flowers. And this is last one is the modern concept. So what is modern concept? So according to modern concept, there are number of pathways such as autonomous pathway, gibberellic acid pathway, photoperiod pathway, vernalization pathway, and environmental pathway, which are integrated to each other and regulate the flowering process. So friends, this is about the theory of flowering. So in this we covered this 7 theory, and in this lecture we will focus mainly on the modern concept. Now come to the first, that is circadian rhythms. So what is circadian rhythms? Circadian rhythms are also known as the diurnal rhythms. They are also known as diurnal rhythms. Also, it is known as internal biological time keeper, also known as the endogenous oscillator, means periodic fluctuations. It is also known as internal and the name is given by the French Hallwork in 1950. So French Hallwork coined the term circadian rhythm. So what is the meaning of this? Is Latin word. The meaning of that, circa diams or dives. Circa means about and diams means days, about day. So what is the definition of this circadian rhythms? So organisms are normally subjected to a daily cycles of light and darkness in both plant and animals. So this circadian rhythms occurs in both plant and animals. and often exhibit rhythmic behavior in association with these changes. Okay, so this is about the your circadian rhythms. Then what are the important characters of this circadian rhythms? So first character is time to complete one cycle in approximately 24 hours. So this is the first characteristics of circadian rhythms. It takes approximately 24 hours to complete a cycle. And second character is they are endogenously generated and they are self-sustaining. So what is the meaning? Means they are internal pacemaker or endogenously generated, means the internal molecules or some genes regulate this process, and self-sustaining means they set and reset according to the environmental situations. Then what is the third characters? They have temperature sensation, and this is given by the Phyton Drick in 1954. So what is the meaning of this temperature compensation? Means this circadian rhythms remain constant under different temperatures regions. So meaning is that they were not affected by the temperature changes. And in the lab conditions, when the organism grown, they show the diurnal variations in the normal condition. But when they transferred into the continuous dark or light situations, they also maintain their rhythms. This is known as circadian rhythms. Means even in the condition of the changes condition, they maintain their rhythms. So they are known as circadian rhythms, and also they have some biological clock that is also known as Jeter Gerber, which reset the biological clock of this. If we give a light before the advance phase of the clock, it will give light after the dusk. It delayed the cycle, and if we will give light during the afternoon or daytimes, then there will be a no effect on this circadian process. Now we will discuss about the, if we will see that time and clock out, it is like that. Okay, so this is known as peak. Period is the time to complete cycle. So this is the maximum or known as peak or maximum, and this is the minima or minimum point of a output, also known as drop. So what is the period? Period is the time to complete a cycle from your peak to peak. So it completes one cycle. Okay. Then second one is the, what is phase? So what is phase? Phase is the time of day for any given event. So if you will see that this is the phase, means any given events. So here what is the position of your output? So this is here. If we will say this is maximum, this is minimum, and this is some middle, so this is known as phase. Then third is the amplitude. So what is amplitude? So it is the 1/2 of the cycles, means from maximum to minimum. This is known as your amplitude, or minimum to maximum. This is also known as amplitude. The description of circadian rhythms. So there will be a three modules. What are these? This is, first one is the input pathway. Second one is the central oscillator, and third one is the output pathway. In the input pathway, we includes the light and temperature. So these are the input pathway of circadian rhythms, which entrance the signals. Okay. Then second one is the central pathway that generate the circadian rhythms. So this is the central or core pathway which generate the circadian rhythms, and third one is the output pathway. So this include the response of these different physiological functions in response to the circadian rhythms. So these circadian rhythms regulate different physiological processes in plants, like plant phosphorylation. Second is the gene expression of flowering and germination. Many genes it regulated. Third, it regulates the flowering process by regulating constant FT and so many genes. Then it regulates the chloroplast movement. It also regulates the level of calciums. It also regulates the stomatal movements, means opening and closing of stomata. It also movement the leaf movements, opening of petals, and the recently they are found to regulate the biotic and abiotic stresses. So these are the pathway that how the input pathway, there will be a light and temperature which interact the signals, central pathway which regulate or generate the signals, and third pathway which is the response to the this signals. So this is the pathway of circadian rhythms. Now we come to the molecular understanding of the circadian rhythms. So friends, there are number of genes which are involved in the circadian process, and we will discuss the some important genes. Like first is the CCA1. This is known as circadian clock associated one gene. Second, this is the homologous of CCA, which is late elongated hypotile. So this is also one. So this gene are expressed during the morning hours. This is known as morning expressed genes, and these are MIB transcription factor encoding. Okay, then second one is the TOC1, which is also known as timing of cap expression. And this gene is the evening expressed gene, and this gene also related with the PAR, pseudo response regulators. So there are three PAR genes which are associated with TOC. This is PAR 5, 7 and 9. So these genes with TOC1 form the central core, and also the gene CRY, which is known as gene for cryptochromes, and this is blue light dependent photoreceptors and promote the flowering. And next one is the PHY. This is also photoreceptors in response to red and far red lights and interact with the ELF 3 of evening complex. Okay. Then there will be one gene that is known as GI. Gigantia. So this gene is acts between your circadian rhythms and photoperiodism. So this gene acts as a between this. Then there are like ELF3 and 4 and UX, that is also known as phyto clock 1. This gene form the ELF3 and Lux form the evening complex of circadian rhythms. And these are the genes. So this is the pathway that how the circadian rhythms, the molecular model for the circadian rhythms. So friends, as discussed, this is the CCA1 genes or leafy genes, which are known as morning expressed gene, and this is the TOC1 and PR5 is the core or central regulated associated with the PR5s. So these genes, this core central regulate the both your evening complex. This is evening complex made up of LUX, ELF3 and ELF4. And this is the genes RBE and LNK, which is known as Reilly and Reilly light regulated, and LNK is the night regulated. So this gene positively regulated your both central core and your evening complex. So activation of this central core inhibits the morning express gene during the evening hours, and this RBE and link gene activates your evening complex. This is Lux, ELF3 and ELF4 during the evening times. And this is the regulation that this morning express gene also inhibit the your evening complex. So this is the signaling that how the circadian rhythms regulated during the morning and evening hours. So in this there are so many genes, which are some genes are expressed during the morning hours like CCA and LHY, and some are the central core which regulate both morning and evening complex, and evening complex, and this and the both morning and evening complex form the your negative feedback loop, means replace the morning genes and evening genes replace the morning genes and morning genes replace the evening genes, and this RBE and LNK, Reilly and night regulated genes form the positive feedback loops. Positive feedback loops which promote the central core and this evening complex. So friend, this is about the your circadian rhythms. So in the circadian rhythms, we comes to know that the circadian rhythms are internally regulated, and there are regulate some important process like photosynthesis, your chloroplast movement, stomatal movement, your petal movement, abiotic and biotic stresses, and there are number of processes which are regulated by the this circadian rhythms. Also metabolism. So there are number of genes like photosynthetic, like Rubin's cage activity, which are regulated by the circadian rhythms. Okay. There are some genes. There are number of genes like CCA, LHY, your TOC1, your RB, your LNK, GI, ELF3. There are number of genes which regulate this input, central and output pathway of your circadian rhythms. So this is about circadian rhythms, and in the mains they can ask one question from this. Now come to the second pathway, that is known as photoperiodism. Sorry. So what is photoperiodism? So photoperiodism is the ability of an organism to detect day length or developmental response of plant to the relative length of light and dark period. So what is the meaning? This is photoperiodism. Photo means light, periodism means periods or duration. So meaning that light duration control the developmental processes in plant is known as photoperiodism. So based on this, plants are classified into the three categories. And in 1920, W. Garner and H. A. Allard discovered the photoperiodism phenomena in mutant of tobacco. And what is the this is the classification of plants. But leaf is the site of perception of photoperiodic signals. So leaf is the site of perception of photoperiodic signals. And plants are classified in HDP, short day plant. Second is LDP, known as long day plant. And third is the day neutral plant, DNP, day neutral plants. So this short day plant is also known as long night plants. It is known as short night plant. So what is the meaning of that? The meaning is that this short day plant required more night time. They required in this plant the flowering process is accelerated if we give more night period or duration than critical day length. What is the critical day length? So critical day length is the period of the light which is crucial for the different physiological process, and if we will give the less duration less than the critical day length, then the flowering is promoted during the short day plant, and if we will give more light than the critical length, then the flowering promoted in the long day plant, and the day neutral plant they are not affected by the this critical period of light, means they are photo insensitive. So generally your kharif crops which are growing during the monsoon season or June to July month, they are generally they are short day plant, and the crop which are growing during the Rabi season like wheat, oat, barley, they are considered as a long day plant, and the crop which are grown during the your summer season, generally they are day neutral plant, or these plant can be grown in any season. So there are some examples, and there are two types. One is the quantitative, and second one is the quantitatives. So what is the meaning of quantitative? Quantitative meanings, sorry, this is qualitative. The meaning of qualitative means they can grow in the only in the short day conditions. They cannot be grow in the long day conditions. And what is the meaning of quantitative? They can grow in the long day conditions also, but the flowering process or flowering phenomena is less. They cannot be grown. These group of plant cannot be grown under long day conditions, or they are known as absolute SDB short day plant, and these are known as facultative short day plants. So some example like coffee, rice, gentium, kalanchoy, hibiscus, glycine max biloxi, this is variety, and Maryland mammoth. They are qualitative STP short day plant, means they only can grow under the short day conditions, and SDB quantitative means they generally they grow under the short day conditions, but if they can grow under long day condition, the flowering process is less. Like that, example is glycine max, sesamum, cannabis sativa, rice, gossipum, cucumis, some species, giamage and sorghum. Long day plants. This is also classified under two groups, that is quantitative and qualitative. And example is alfalfa, pea, petunia, potato, wheat and barley, Arabidopsis. And qualitative, Nicotiana sylvestris, papaver, trifolium, Niger, coriander, brassica, juncea. So friends, this is the example of short day plant, long day plant. And, in day neutral plants, there are like tomato. Tomato is the day neutral plant. Also, corn, cucumber, sunflower, they are grouped under the corn, cucumber, sunflower and tomato. They grouped under the day neutral plants. Now come to the mechanism regulation of flowering by photoperiodism. So in this also the timing mechanism of flowering is based on the endogenous circadian. So this circadian rhythms contain two half. One is the photophil, and some second one is the scotophil. This is the light period, or it is known as dark period. During this phase, the flowering is promoted in the long day plant and vice versa. And two photoreceptors like phytochrome and cryptochrome, they regulate the photoperiodism process. Then there will be one hypothesis that is known as hypothesis. So according to this hypothesis, 12 hour long, there are 12 hour of photophile and 12 hour of scotophile phase. And when we will light during the scotophase, the flowering promoted in the long day plants. And second character is the regulated by the endogenous circadium given by the Witten, Drake, Dorothea in 1964. That photoperiodism is regulated by the endogenous circadium, or this is known as internal coincidence model. Means circadian clock this reset the photophile and scotophile phases. Now how this photoperiodic regulations take place. So we will discuss about the photoperiodic regulations. So in this also the genes like CCA we have discussed in the circadian rhythms. This gene and its homologue left hypocotyle bind to the promoter of TRR5 pseudo response regulator 5 and one gene that FKF known as flavin, winding, calch, wheat, this is known as FKF1 and GI, that is gigantia. Already discussed about this gene. So this bind to the promoter of these genes and repress their expression. This PRR5 are negatively control the CDF genes. What is CDFs? Cycling dwarf factor. This is a repressor the constant gene, and constant is the fluoring genes. So this FKF1 and GI complex regulate the activity of this, degrade the CDF genes, and there will be an expression of this genes which regulate the fluoring process. So what happen in the how this photoperiodic regulation take place? So in these two genes, which are circadian regulated genes like CCA and leafy, which bind to the promoter of these three genes PRR5, FKF1 and GI, and this FKF and GI and this bind, and this PRR5 represents the expression of CDF genes, and these CDF genes are the repressor which are bind on the constant genes, and constant are the lower genes. Also the FKF1 and GI degrade this CDF genes, and there will be an expression of constant genes. Also some proteins which activate the further this cogene activate the FT genes, and FT genes cause for the fluoring process, and some genes like COP1 is also activate the active protein. So this is the regulation of photoperiodic in which some circadian genes bind to the promoters to the PRR5, and PRR5 repress the CDF genes, and FKF1 and GI this degrade this repressor CDF genes on the constant, then later on expression of active genes, and some co-proteins which are also activate active protein, that is known as COP1, which so this is the photoperiodic regulation of the now there will be also photoregulations in the wheat and barley. That how this wheat and barley, these are the long day plants. Earlier we have discussed about the Arabidopsis, a model plants. So here there will be a phytochrome C. It regulates, activates the PPD1s, and this PPD1 is homologous to the PRR7 pseudo response regulator, which is known as photoperiod 1, and this gene activate the FT1 genes and more the fluorine. Also this phytochrome C activate the circadian clock. Discuss there are number of genes CCA, PRRs, GI, LUX, ELF3, and they activate the constant 1 and 2 and activate the fluoring locus T genes. Also, this is the crosstalk of the photoperiodism and circadian rhythms. Then there will be a process that is vernalization. So this vernalization, there is a gene which is activate the VRN1, and this VRN1 activated the VRN2, which is act as a fluoring repressor and inhibit the FTI genes. Expression of this FTI genes activate the VRN1. So this is the regulation of this photoperiodic pathway and crosstalk of vernalization and circadian clock in the regulation of fluoring in the your wheat and barley. There is also one that how the fluoring induced in the your short day plant, like model plant is rice. So in rice there is a one gene that is OS, means Oryza sativa. GI is our circadian rhythm genes, that is Gigantia. It activates the HD1 gene, and this HD1 inhibits the 3A genes. However, in the presence of red light, cytochrome V, and HD6, it activates these genes. Also, they regulate the genes, that is ESD1, activate the LEFT1 FT1 gene, and it induce the fluoring. Also, they regulate one transcription factor, that is MEDVOX, which activate the genes ESD1, or directly they can activate the ESD1, which activate this SD31 genes. ESD31 also activate this. So this is the regulation of fluoring in the rice. So like this, the photoperiodic process are regulated in the short day plants and long day plants. So this is also a important process in the regulation of your fluoring process. Now come to the third pathway, that is very important, thermoperiodism. So FW Wendt in 1944 described a phenomenon in which growth and development is promoted by the altering day and night temperatures. So thermoperiodism form potato tubers in response to the low night temperature, and also fruit set in tomato, which is activate the fluoring by the low night temperature. There are some cardinal temperatures. What are these? So this is the plot for the any response. This is any response or physiological process, and this is temperature. This is known as zero is known as base temperature. temperature below that there will be a no growth. So this is known as base temperature, and this is the minimum at which the any response is lowest, and this is known as maximum at which the process is highest, then optimum at which the process work efficiently. So these are the cardinal temperatures. About the, as you know that different tissues, different tissue within same plant have different cardinal temperatures. Even the cardinal temperatures are vary with the stage of the plant growth. Means for germination there will be a different temperature requirement. For vegetative growth there will be different. For stem growth, root growth and leaf growth there will be a different cardinal temperatures. For better fitness of plant, the temperature should be near optimum. Okay, friends. Also, the above ground temperature and below ground temperature, that is known as soil temperature, also different for root and shoot tissues. Now come to the mechanism of this thermoperiodism. So recently there are some thermo sensors are discovered which responds to the different regimes of temperatures. Means if we will give different regimes of temperature to the plant, they are activated. Like some RNA, although they are not fully characterized. So here that PIF7, it is a stem loop structure, act as a thermosensor. Then one protein, that is ELF3, act as a thermosensor, and you know that this gene also involving your circadian rhythms, and some protein metabolites like phytochrome B and PHOT. These photoreceptors also sense the thermoperiodism. Now there will be one experiment. This event conducted a experiment in tomato. What he observed that if we kept a plant in a constant 26 degree centigrade or constant 80 degree centigrade, then there will be a very poor growth. Also, there will be a failure of failure in flowering and fruit setting. Then they gave the alternate 26 degree at daytime and 80 degree centigrade during the night time. Then they found there will be a vigorous growth. And when they oppose this temperature, means 26 degree during your night time and 18 degree centigrade during daytime, then there will be a also failure in the or very low poor growth. So from this study we can say that plant need a variation in the day and night temperatures, and the night temperature is more important than the day temperatures. And this thermoperiodism is very important for the temperate area, and where we use this glass house or greenhouse industry, which can increase the sustainable growth and development. So friends, this is about the thermoperiodism. So we can say that plant sense the temperature changes, and they need the diurnal variations or different regions of temperature. Like they have some maximum temperature, minimum temperature, base temperature, and some optimum temperature. So wetter fitness, we need a wetter means optimum temperature. Also, there are some thermosensors which are identified but not fully characterized, like PIF7, ELF3, and phytochrome B and phototropin. The phytochrome B and phototropin are photoreceptors. These sense these changes in the temperatures, and there will be one experiment conducted by the vent which shows that plants needs a variation in temperature, and generally the day temperature will be high than the night temperature. So this is about the thermoperiodism. Now come to the vernalization. So what is vernalization? So vernalization is the induction of flowering process by the exposure to the prolonged cold or winter. So this is known as vernalization, and the one scientist that is known G, given the name vernalization. And this vernalization causes the tense same. So we have discussed already that what is the competence word. So word which are ready to convert into the juvenile phase to the vegetative if proper photoperiods and hormones are provided. Then this phase converted in then word converted into a determined word which go in the future is that flower production. And the effective range for this vernalization process is between 1 to 7 degrees centigrade and 10 degrees centigrade, and ranges is 1 to 7 degree, means below which there will be no growth. And the most common combination for the cold treatment plus long day which promote the flowering process. This is about the vernalization. And many monocarpic tree plants and biennials need vernalization process for flowering. And in the vernalization, it involves the epigenetic changes. So what is the epigenetic changes? Epigenetic changes is the stable changes in the gene expression without involved alterations in the DNA sequence. So they are the permanent change or stable change in the gene expression without involving the change in the DNA sequence. Means this cell go through the mitosis and meiosis. And flowering locus C, we know that this is flowering repressor. This FLC highly expressed in the non vernalized stem. And also 3 genes which are involved in vernalization process: BRN1, BRN2 and BRN3. And this BRN2 act as a repressor, and this flowering stimulus is transported through the phloem. Now come to the mechanism of this vernalization process, that how this vernalization process take place. Already discussed that FLC, this is a major flowering locus C, is a major regulatory of vernalization. And VRN1 and 2 found in diploid wheat, Triticum, coccum, associated with vernalization process. In barley, that SSG2, spring growth habit. And in rye, SP1, related to spring growth habit. This gene causes for the vernalization process. So these are the genes in wheat, barley and rye associated with the vernalization process. And this gene encode a zinc finger domain, that is known as CCT, constant co-like torque 1. So they regulate this gene finger. In Arabidopsis, there will be a one gene, that is FRI, known as Frigida, which activates the FLC gene, flowering process in Arabidopsis. Also the vernalization process, which is also inhibit the FLC genes, and the VRN gene, VRN first, also which activate the flowering process. So this is about. Also there are some points. Related to vernalization, like where this vernalization process perceived by the shoot apex. So these vernalization signals are perceived by the shoot apex. Hence, there will be a epigenetic changes take place. Activate the FLC gene, which act as a repressor of fluorine. Then VRN2 repress the fluorine under long day condition by inhibiting FT genes. Also inhibited by BRN1 and 3 is equivalent to fluorine locus T in the Arabidopsis. This vernalization response can be transmitted through the grafting. So this vernalization is used for the conversion of winter varieties to summer variety. So this is the signaling mechanism that shoot apex this receive the vernalization signals, and there will be a mitosis meiosis take place. And there will be epigenetic changes in the genes, activate the FLC gene which is repressed, and this is about signaling process. So this is about the vernalization process. Next, there will be a GA signaling, and GA is the hormone. And you know that in Florigen theory, the hypothetical molecules which is given by the Chelakian is the gibberellin plus anthycin. So how this gibberellin signaling regulate the flowering process? So there will be a GA hormone which regulate the some nuclear factors like receptor DELA, GAI, RGA, RGL 1, 2 and 3. So this gibberellin encode the nuclear receptors DELA, GAI, RGA, RGL 1, 2, 3, which inhibit the GA might be genes. Also, they inhibit the mirror microRNA 159, which further repress this. And in the presence of sucrose, it activates the SOC1, which remove these repressors DELA of this and activated the leafy genes. And these leafy genes promote the flowering. So in the presence of gibberellic acid, this bind to the nuclear receptors, and binding to these receptors for the degradation of these receptors, which are act as a repressor, and there will be activation of genes. This is gibberellic MIB genes, and it's activate the attached to the leafy genes, which promote the flowering process. So this is about the gibberellin signaling. Now come to the next, autonomous pathway. So already discussed that in autonomous pathway we include mainly photoperiodism and your circadian rhythms. This autonomous pathway appear to regulate flowering independently of environmental cues. In very expressed repress FLC expression. Some genes like FCA, FY, FLD, FPA, LD, FLK are associated with this autonomous pathway. Now come to the next, which is environmental regulation of this flowering pathway. So this is also one of the important pathway which regulate the flowering. So you know that these plants shows the plasticity, means as environmental change, plant also try to change according to the environment, and there are different interactions and variations like diurnal changes, seasonal changes, annual changes, or long-term changes of environmental take place. So first factor is the latitude. So you know that in the pole and equator. So equator, the day and night length is almost equal. So in this condition mainly they are these day neutral plants are dominant. But in another case, there will be a variation in the light, and so there will be a short day plant and long day plant. So means according to different the plant composition is also different. So means geographical latitudes is also affect the flooring. Then light spectrum. You know that light is also one of the important factor to regulate the flowering process. And your phytochrome, cryptochrome, which respond to the different light. Like blue light, for blue light there is cryptochromes, and for red and far red lights there is a phytochromes. And also according to this, your short day plants, long day plants, and day neutral plants, and the light period photoperiodism also affect the flowering in short day and long day plants. So we can say that light affect the then shading. So in one experiment, when the light was reduced by 37%, 25%, and 11%, then there will be a reduction in the 40, 56, and 79% in the flooring process. So this study can say that shading also affect the your flooring process. Then water, you know that both that excess and low water both pose a stress for plants, and it can accelerate the flooring. That's why there will be a different plant like drought escapers, which induce the flooring in the stress condition. Then what are other factors? Like already discussed, CN ratio, that increase carbon and nitrogen ratio promote the flooring. Then in some experiment, application of nitrogen is also induce the floor. Then temperature, we already discussed in the thermoperiodism, that thermoperiodism and vernalization, that different plant have different cardinal temperatures which regulate the growth, development, and flowering process. Then day length, then high temperature reduce the flowering. Environmental stress. So there are so many stress like salinity, alkalinity, acidity, high temperature, drought, flood, which also affect your flooring mechanism. So these are the pathways which affect the flooring process. So friends, what's modern theory says? So in this lecture we have discussed that there are so many pathways like then autonomous pathway, then gibberellic acid pathway. So this vernalization pathway have some genes like BRN1, 2, 3. And autonomous pathway has some gene like FCA, FY, FVA, FV. So these two pathways need the FLC, which act as a lower the pressure in the inflorescence meristem. Inflorescence meristem. This pathway inhibit the FLC vernalization. This gene further inhibit the SOC1. And there is a one pathway that is called as photoperiod pathway. Photoperiod pathway. In this pathway, the long day conditions induce the GI gene, Gingatia, which promote the constant flowering genes in the presence of phytochrome A, and this gene activate the flowering locus T mRNA, which move to the meristem cells. And FT protein with the FD and form the FT or FD complex, which activate this SOC1. And this SOC1 activated the leafy genes. Further, they promote the reversibly patella genes and regulate the floral genes. And also one pathway, that is gibberellic acid pathway, which activate the SOC1, which we already discussed, and also it activated the leafy genes. One is also activated these leafy genes. And one more pathway, that is age pathway, which activate the microRNA 156, inhibit the S, and promote the, and that inhibition of these genes activate the leafy genes. And also this activated these leafy genes and microRNA 172 inhibit the patella 2 and also inhibit the SOC1 and also inhibit the FT. So this is the combined pathway. So friends, in this we can see that there are four pathways which are involved in the modern flowering pathway. And first is the vernalization pathway, and we already discussed that there are three genes for regulation of vernalization: BRN1, BRN2, and BRN3. BRN2 act as a repressor. So this gene represents the FLC gene, flowering locus C gene. Then second pathway, autonomous pathway. In that, the genes like FCA, FY, FVA, and FB also regulate this FLC gene. Then there will be a one pathway, that is photoperiod pathway or long day pathway, which induce the formation of this Gingatia genes in the presence of phytochrome A and induce the expression of co-genes, constant gene, which induce the expression of FT-mRNA, and this FT-mRNA move to the meristem, form the FT protein, and this FT protein attach with the FD gene, and this FD induce the formation of this SOC1. Okay, and this SOC1 induce the formation of leafy, which form the floral meristem genes. and also this leafy this reversely regulate the apatella 1 genes. And one in another pathway, this gibberellic acid pathway, it activates the SOC1, and then SOC1 activate the leafy genes, and leafy genes is regulate the your fluoring pathway. Then there will be one pathway, that is ACE pathway, which activates the microRNA 156, which inhibits the SPL genes, and this SPL genes activate the leafy genes, and also this activate the microRNA 172, which inhibit the apatella 2, which further reduce the SOC1 and inhibit the fluoring process. So this is about the modern fluoring pathway, which regulate the fluoring pathway. So in this lecture, we discuss a complete developmental biology of fluoring, in which we discuss that which are the fluoring pathway, given earlier, and what is your circadian rhythm. We read about its properties, characteristics, functions, and their signaling mechanism. Then we have discussed about the photoperiodism, that how the plants are classified, signals, how they induce fluoring or their role in fluoring. Then we discuss about the vernalization pathway, that how this vernalization pathway regulate the uh, fluoring process. Then we discuss about the thermoperiodism, that how the temperature enzymes are responsible for periodism. Then we discuss about the gibberellin signaling. Then later on we discuss about the autonomous pathway. Then we discuss about the environmental cues for regulation of fluoring. And at the end we discuss about the integration. We integrated all the process in a one fluoring pathway. So friend, this is a very important topic for all the competitive exams, and hope you will understand this lecture. Thank you.